So now we're going to talk about the favorite part of my protein discussion, determining the primary sequence of a protein or a peptide. So there are 6 steps in this process. We're going to start obviously with step 1. If, and only if, we have quaternary structure with our protein, then we have to separate the chains. So remember, in that quaternary structure, those subunit interactions are based on weak IMFs. So we can separate them using extreme pH, whether that's alkaline or acidic, 8 molar urea, 6 molar guanidine HCl. So this would be basic, this is acidic, or high salt concentration. Usually the substance used for that is ammonium sulfate. So what if we have a polypeptide chain that contains disulfide bridges? We're going to have to break those bonds between those sulfur atoms, and there are 2 methods for achieving that. One is an oxidative cleavage, the other is a thiol modification. So we're going to look at both of those. In the oxidation, the oxidizer is performic acid, and basically we are going to add oxygen across that single bond and break it. So we get this SO3 minus group on the sulfur. This prevents the reformation of that disulfide bond. So it's a one-step done. There is another method, and that's the SH modification. But in order to modify this with the 2 substances shown here, iodoacetic acid or 3-bromo polypropylene, you have to still first break that sulfide bond. And what we use for that are sulfhydryl compounds like 2-beta-mercaptoethanol or dithiothriotol, DTT. These reduce those disulfide bridges in the cysteine residues, but they leave those cysteines free to reform that bridge. So you must treat them in a second step with an alkylating agent. So that's what these 2 compounds are doing. They're alkylating that sulfur so that disulfide bond cannot reform. So that's the SH modification. We need to identify N and C terminal residues. So remember that polypeptide chain has an amino terminus and a carboxyl terminus. So we've looked at Edmond reagent. I've already told you that identifies N-terminal amino acids. So remember, Edmond's reagent is phenyl isothiocyanate, and it produces a derivative, a PTH, connected to the amino acid, and then that PTH derivative, or that product, is then analyzed in order to identify what amino acid is there. We can also use, and I don't think it's on this slide, nope, so I'm not going to I'll stay here for a minute, carboxypeptidases. And these will cleave the C-terminal amino acids. And then we can isolate that amino acid and identify it. So if we use repeated rounds of Edmond's degradation or reagent, we can identify a number of residues in order from the N-terminus of that amino acid. You've seen this before. This is just another representation of that reaction with the phenyl isothiocyanate. And again, we're not going to go through this step by step because I don't want you to worry about the real details here. But again, be able to recognize that PTH amino acid. Other information about Edmond degradation. You can usually identify up to 50 amino acids from the N-terminus if that polypeptide is 100 to 200 amino acids long. However, the efficiency of this process when the polypeptides get larger, so we're talking about in the thousands, we can typically only get 10 to 20 of the N-term amino acid sequence from this process. So we've identified N-term, C-term amino acids, hopefully, by this point. The next step is to fragment that chain, and we use enzymes to do this. Trypsin, chymotrypsin, clostripan, staphylococcal protease. We can also chemically fragment using cyanogen bromide. So how does this work? We're going to take a look at enzyme fragmentation here. We're using the enzyme trypsin. Trypsin cleaves peptides with C-terminal arginine or lysine residues. So what that means is that it's going to cleave the peptide on the C-terminal side of lysine. So again, remember, when we write amino acid sequences, we start at the left, we go to the right, that's the way we read them, and turn to C-term. So this is the C-term side of lysine. So it cleaves C-term of both arginine and lysine. Here we have an arg, here we have a lys, we have another lys, and there's another arg. So when trypsin fragments this polypeptide, we are going to get 5 fragments. Hope that makes sense. So if we have 4 residues that are targeted by trypsin, we'll get 4 fragments plus 1. Okay? So you ought to be able to come up with a number of fragments that will be produced by enzymatic cleavage based on the number of those amino acids in the peptide chain. Cyanogen bromide, chemical fragmentation acts only on methionine residues. So this is pretty useful because proteins usually have only a few of these, and that will help identify their location. We're able to recognize the results based on a peptide with a C-terminal homoserine lactone. Here's a representation of that reaction. So here we have a methionine residue. There's a nucleophilic attack of the methionine sulfur on the carbon atom in that cyanogen bromide. So this is attacking here. We release the bromine or the bromide ion. And then in the second step, we're going to release methyl thiocyanate, and then at the C-terminal of that fragment, we're going to have that homoserine lactone, which can be identified. So here's a table that contains some of the more common enzymes and chemical methods that are used in fragmentation and sequence analysis. Trypsin, chymotrypsin, clostripan, staphylococcal protease, which I mentioned. This indicates what side of the amino acid, whether it's the carboxyl side C or the amino side N. You'll notice that these all cleave C-term to that residue, and these are the residues that are targeted by these enzymes. As I said, arg-lys for trypsin, chymotrypsin, phenylalanine, tryptophan, tyrosine, leucine, clostripan, arginine, staphylococcal protease, aspartic acid or glutamic acid. N2OH will cleave asparagine glycine bonds, and this occurs at a pH of 9, so it is pH dependent. At pH 2.5 and a particular temperature, so this has to be warmed because this is higher than room temp, it will cleave aspartic and proline bonds within that polypeptide chain. So depending on pH, that NH2OH is selective with respect to its hydrolysis. The final step then is going to be reconstructing the sequence. And what we're going to do is basically match those fragments up. So we're going to have a number of different fragments. We're going to sequence all these fragments, usually by Edman degradation. We might use another method that we'll talk about momentarily. And then we'll compare and align the overlapping sequences to obtain the sequence of the original polypeptide chain. And I'm going to talk about mass spec for just a few minutes, and then we're going to take a break, and we are going to actually work some of these types of problems. I think they're fun. Think of them as like a puzzle, and hopefully you'll have fun working them too. So one way to analyze or determine sequences for polypeptides as well is the use of mass spectrometry. And what we have here is a tandem mass spectrometer. It's an electrospray format. And this is a relatively new method, became more usable in the mid-80s, about 1985. And what this method does is accurately measures the mass to charge, or M to Z, ratio for ions in the gas phase. So in this process, we have to evaporate and ionize the molecules in a vacuum, and then they are separated in space and or time based on the mass to charge ratios as they go through the mass spectrometer. And the spectrometer at the detector end is going to measure the abundance of ions with specific mass spec ratios. Okay. There are a couple different formats for this. This is electrospray here, so rather than a gaseous phase, we're using a solution that's sprayed. So these fine droplets are going to be charged and travel down that mass spectrometer to the detector. Another very common and accurate method is using what's called matrix-assisted laser desorption time of flight, or MALDI-TOF. It's not pictured here, where that sample is then mixed with a chemical matrix, and you spot it on a metal target, and a laser pulse ionizes the sample and ejects it into the gas phase, after which the ion fragments are run down through the mass spectrometer, hit the detector, and then analyzed based on mass to charge ratio.